Hello everybody and welcome to Biology Doodle, and today we will be going through glycolysis. Glycolysis harvests chemical energy by oxidizing glucose to pyruvate. Glucose means sugar splitting, and that is exactly what happens in this pathway. So glucose, a 6-carbon sugar, as is seen in the red dots, is split into 2 3-carbon sugars, pyruvate, seen down at the bottom. So glycolysis has 2 stages: the energy investment stage from glucose down to glyceraldehyde 3-phosphate, that is from about here to here, and also the energy payoff phase from glyceraldehyde 3-phosphate down to pyruvate, that is from here down to here. So glycolysis can happen under aerobic or anaerobic conditions. Aerobic means with oxygen and anaerobic means without oxygen. Glycolysis also happens in the mitochondria of the cell. So let's get stuck into it. In step 1 over here, hexokinase transfers a phosphate group from ATP to glucose, making it more chemically reactive. The charge on the phosphate also traps the sugar into the cell. So all of the enzymes that are used within glycolysis can be seen in purple. Step 2. Glucose 6-phosphate is converted to fructose 6-phosphate. This is an isomerization step and is happening here by the enzyme phosphoglucoisomerase. Step 3. Phosphofructokinase transfers a phosphate group from ATP to the opposite end of a sugar, investing a second molecule of ATP. This is a key step for regulation of glycolysis. Step 4. Aldolase cleaves the sugar molecule into 2 different 3-carbon sugars, known as glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Step 5 is the conversion between DHAP and GAP. This reaction never reaches equilibrium, and GAP, or glyceraldehyde 3-phosphate, instead moves on to the next step and is used up. Step 6. There are 2 sequential reactions in this step. Glyceraldehyde 3-phosphate is oxidized by the transfer of electrons to NAD+, forming NADH. And also, using energy from this exergonic redox reaction, a phosphate group is attached to the oxidized substrate, making a high energy product. Now, if up until now some of these words have been confusing, like oxidation, redox reactions, kinase, etc., don't worry, because all of the definitions will be seen below in the description. Step 7. The phosphate group is transferred to ADP in substrate level phosphorylation in an exergonic reaction, and the carbonyl group of glyceraldehyde 3-phosphate has been oxidized to the carboxyl group of an organic acid known as 3-phosphoglycerate. Step 8. The enzyme phosphoglyceromutase relocates the remaining phosphate group from 3-phosphoglycerate to 2-phosphoglycerate. Step 9. Enolase causes a double bond to form in the substrate by extracting a water molecule, yielding phosphophenol pyruvate, or PEP, a compound with a very high potential energy. And lastly, step 10. The phosphate group is transferred from PEP to ADP, which is a second example of substrate level phosphorylation, overall forming pyruvate. So now let's look at the energy payoff. So let's go back to the start. 2 ATP were used in the energy investment phase. And then in the energy payoff phase, 4 ATP were formed, 2 NADH and 2 protons were formed, as well as 2 pyruvate and 2 water molecules. So that is a net production of 2 pyruvate and 2 H2O, a net production of 2 ATP and a net production of 2 NADH plus 2 protons, which will go on to the Krebs cycle and will be used later on to make more high energy compounds and to release more energy into the cell. Okay, so thanks for tuning in to our YouTube video today about glycolysis. Don't forget to like and subscribe to Biology Doodle, and have a look out for our next video, which will be on the Krebs cycle, which is the follow-on in aerobic respiration, and which will cover the transfer of pyruvate ore to acetyl-CoA and how that will then move on to create more ATP and release more energy into the cell.